A new artificial enzyme has shown it can chew through lignin, the tough polymer that helps woody plants hold their shape. Lignin also stores tremendous potential for renewable energy and materials. Reporting in the journal Nature Communications, a team of researchers from Washington State University and the Department of Energy's Pacific Northwest National Laboratory showed that their artificial enzyme succeeded in digesting lignin, which has stubbornly resisted previous attempts to develop it into an economically useful energy source. Lignin, which is the second most abundant renewable carbon source on Earth, mostly goes to waste as a fuel source. When wood is burned for cooking, lignin byproducts help impart that smoky flavor to foods. But burning releases all that carbon to the atmosphere instead of capturing it for other uses. The researchers say that their biomimicking enzyme showed promise in degrading real lignin, which is considered to be a breakthrough. They think there is an opportunity to develop a new class of catalyst and to really address the limitations of biological and chemical catalysts. Lignin is in all vascular plants where it forms cell walls and provides plants with rigidity. Lignin allows trees to stand, gives vegetables their firmness, and makes up about 20 to 35% of weight of wood. Because lignin turns yellowish when exposed to air, the wood products industry removes it as part of the fine paper making process. Once removed, it is often inefficiently burned to produce fuel and electricity. Chemists have tried and failed for more than a century to make valuable products from lignin. That track record of frustration may be about to change. In nature, fungi and bacteria are able to break down lignin with their enzymes, which is how a mushroom-covered log decomposes in the forest. Enzymes offer a much more environmentally benign process than chemical degradation, which requires high heat and consumes more energy than it produces. But natural enzymes degrade over time, which makes them hard to use in an industrial process. They are expensive too. It's really hard to produce these enzymes from microorganisms in a meaningful quantity for practical use. While researchers have been unable to harness natural enzymes to work for them, they have over the decades learned a lot about how they work. A recent review article by the research team outlines the challenges and barriers toward the application of lignin degrading enzymes. Understanding these barriers provides new insights toward designing biomimetic enzymes. In the current study, the researchers replaced the peptides that surround the active site of natural enzymes with protein-like molecules called peptoids. These peptoids then self-assembled into nanoscale crystalline tubes and sheets. Peptoids were first developed in the 1990s to mimic the function of proteins. They have several unique features, including high stability that allows scientists to address the deficiencies of the natural enzymes. In this case, they offer a high density of active sites, which is impossible to obtain with a natural enzyme. As expected, these artificial enzymes are also much more stable and robust than the natural versions, so that they can work at temperatures up to 60 degrees Celsius, a temperature that would destroy a natural enzyme. The researcher says this is a significant step forward in being able to convert lignin into valuable products using an environmentally benign approach. If the new biomimetic enzyme can be further improved to increase conversion yield to generate more selective products, it has potential to scale up to industrial scale. The technology offers new routes to renewable materials for aviation biofuel and bio-based materials, among other applications.